So this is what we call as lab on a chip. Yeah, and actually our lab is one of the lab in Indonesia that is currently working actively in this field, microfluidics. The idea is how can we minimize or miniaturize laboratories. So normally we have to do PCR in large instrument. Now we want to make it done in simple small circuit, like the project done by Ezra and Anjali, yeah? Small scale PCR. So that is comes the idea of micro lab on a chip. So this originates in the 1990s, and they are evolving from the technology developed in microelectromechanical system, MEMS, combined with analytical chemistry and electronics and material science. So in the basic principle of microfluidic chips, the difference is you operate liquid on very small scale. In that case, then from physics point of view, they will have laminar flow because the Reynolds number is very small. Reynolds number is a parameter, a dimensionless parameter in fluid mechanics. Yeah, if the dimension of the pipe tubing is large, then you will have high Reynolds number, where you could have turbulence. The turbulence is the swirling of liquid there inside the pipe. But when you make the pipe very small enough, then you make sure that the liquid will flow in layers within the piping. Where with the liquid layer that attached to the pipe will have zero velocity, while the liquid layer in the middle will have the maximum velocity. Yeah, so if we draw, when the Reynolds number is very small, you have this kind of velocity profile. Okay, this is the middle liquid. The liquid in the middle will have the highest velocity. So this is the factor 2 of liquid velocity. Well, the edge will have the minimum velocity, or maybe even zero, because they are attached to the wall of the pipe of the channel. So this is the characteristic of microfluidic, because the channel is very small, laminar flow. Okay, then oops, sorry. Okay, so the idea is how can we also do similar operation like we do in the big world, yeah, in the PCR world or in analytical chemistry world. We mix samples, we do operation, we heat the samples, and then we do analysis. We do evaporation, then mix between reagents and analytes, etc., etc. How can we do it in a small scale? Yeah, the idea of making it small is, of course, so that then you don't need a large volume of sample. Then you also have the smaller amount of analytes required, which means we lower the cost of the analysis. That is the idea of micro-total analysis system, or lab on a chip, or microfluidic chip. The microfluidic chip itself usually currently is designed using glass material, using microelectronics technology, yeah, with etching, bonding, and then patterning, etc., like that. Although our lab, which I already started since when I worked at UTAS Malaysia, is to try to work it using fabrics-based device. Yeah. So we'll just look. So the problem is that with laminar flow, it is very difficult to have a mix between different flow. For example, like this, then they cannot mix because it is laminar flow. That's why then in order for them to mix, that you have to use special techniques, yeah, various techniques like using what we call like electrostatic mixture. You can use also chaotic mixer and many different kind of techniques. Yeah, so this shows complex flow control, but realized by the surface modification of the microchannel. A, B, C, and D here show the Flow modes achieved by the control of charge polarity located on the inner surface of the microchannel. So this is using electrostatic flow control. Yeah, by putting different charges at the wall, at the inner wall of the channel, you can influence where the flow is going. E and F shows the opposite direction of flow can be realized in a single channel by using applying different charge here. One at the lower part, it flows the liquid flow to the right direction. At the upper part, the liquid flow to the left direction. Yeah, or this is the cross section. So this is using electroosmosis to directly drive the charge flow. It can be also used to pump the liquid, yeah, and we call it electroosmotic pump. Other methods, other materials besides glass, you can also use PDMS, a type of polymer, flexible polymer, polydimethyl methoxysilane, to form a hermetic electroosmotic graphic. In our lab, we use cotton fabrics, yeah, or paper. So this is the case of electroosmotic pump structure. We have a solution 1 here and solution 2, and in electrode 1 and 2 and electrode 3 here to pump the fluid, yeah, from from here to this direction. And the other is using air, what we call is pneumatic micropump structure. So by applying different airflow at different region of the above the vertical above the fluid channel, then we can influence the direction of the fluid flow. So microflow control is the centerpiece of microfluidic chip operation. It is related to almost all the processes such as sampling, mixing, reaction, and separation, which are necessarily to be finished in the controllable flow. So this is the different operation that require to be done in microfluidic flow control, sampling, mixing, reaction, and separation. In fabric-based or paper-based microfluidic device, on the other hand, they do not require pumping because the flow occurs through capillary forces. While in pumping is usually required when you have the microfluidic in hollow channel device. Okay, so this is different ways of valve. Yeah, valve is how to control the flow also, yeah, using silicon cantilever structure, that using electrostatic actuation. This is a normal when this silicon cantilever is attracted to the bottom part, liquid can flow from this left to the right part. When it is touched the upper part, liquid will be blocked. Okay? And this is the passive one-way valve. And sample operation mode, the sampling is required usually when we are applying the microfluidic system for analysis. So the analysis object is first being sampled and stored in the sample storage tube. Then it is introduced to the microfluidic analysis system in the sample storage chamber, and then being pre-treated in the sample pre-treatment system. After that, then it undergoes separation and analysis. So this is one way of using electrical force, yeah, to introduce a sample. And so this is the sample chamber. And number 2 here is the sample waste solution. And number 3 is the buffer solution chamber. And number 4 is buffer waste chamber. So when changing the voltage, yeah, like this, first the sample flow from number 1 to number 2. Then with changing the voltage, yeah, there's a high pressure of buffer coming from 3 to 4. This will push some of the sample here to downward. So we have a droplet of the sample here. Yeah, so we call it droplet microfluidics or digital microfluidics. So instead of dealing with sample in continuous flow, it will deal the sample in this small volume droplets. Filtering is often used in microfluidic system. This is, for example, using microchannel based filter, in which that there is a channel having small gaps between them. And this B is using magnum based filter, and the C here is using micro square cage based filter. So the sample is being stored in the middle, and then only sample which has smaller size, then the gap here can flow through. Then micromixer, reactor, and separator, like this. The design of an electrophoresis chip, when we have the sample chamber here, the buffer chamber here, the waste chamber is here. Then using electrostatic voltage, the sample can be controlled to flow towards this waste chamber, while the compound inside it can be detected. The detection methods in a microfluidic device can be different, can be many different ways. It can be simple as colorimetric analysis, can be using a light and then detector analysis like this. Can also use electrodes embedded into the channel to detect different substances using ion selective electrode or using other chemical sensor application. So application of this is many. Yeah? So because now, as I mentioned, many of the molecular analysis done in biology are still done in using bulk instrumentation, PCR, and then also protein analysis. But more and more companies are building microfluidic solution to this big analysis to make a smaller device. For example, like the newest DNA sequencer, made by ion pore. It uses also microfluidic device combined with pore, nanopore structure to detect the sequence of the DNA. And it is developed by a company in UK, United Kingdom. So when this book is written, it is still with SARS-1 in 2003. So they developed here non-invasive method for SARS virus detection using portable or miniaturized PCR microchip like this. So in the PCR region, the the temperature of this region is being controlled in three different region. The denaturation, the annealing, and the denaturation, the hybridization, and the annealing. And then the detection is using a light source in a microscope, laser and CCD, by laser induced fluorescence system. So the chip is in this region. Detection of amplification is done using laser. So this is actually what I would like you to build, especially Ezra and Angelic Group, to develop a small PCR device but not using glass like this, but instead we use fabric base. For now, your task is only to develop a thermal cycler using fabric base device, not yet to be able to amplify actual DNA. Is there any questions related to microfluidic and lab on a chip or total what do you call it? Total analysis system, micro total analysis system?
Sir,
Yes.
I want to ask, do we have P microfluidic printer buat paper base di lab kita?
What do you mean with
Jadi printer yang bisa wax pakai wax.
No, we don't have wax printer. It is expensive. Yeah. So what we have, I hope you already learned from Elga, maybe the
Yang mana, sir?
Diana and siapa namanya? Elnora already also learn how to use it. So you use digital cutter. So because the digital wax printer is expensive, what we do is you make the pattern using paper, and then you impregnate the paper using wax. And then the wax is transferred from the paper into the fabric using laminating machine. So this is from my paper in 2012. As Chris has read that paper, right? Chris, can you explain a little bit?
Yes, microfluidicnya ya.
Yeah.
Jadi ya di paper itu kita bikin kayak pattern, terus habis itu kita celupin ke dalam waxnya. Nah, terus pattern yang yang channel yang mau dibuat itu tuh kayak kita bolongin gitu, habis itu tinggal kita tempelin ke cottonnya, kurang lebih gitu.
Yeah, so the wax is being transferred from paper to cotton fabrics. Because we cannot directly cut the cotton fabrics using digital cutter, yeah. It's just difficult. It's easier to cut the paper. So the wax is the paper is used as a template to transfer the wax pattern into the cotton fabric. So please ask Grace, the paper. You please read the paper. You can ask also Diana and Eldora. Only for the PCR analysis, however, since you require operation at high temperature, right, until 95 degrees, you cannot use simple wax. Yeah, the previous group, Diana and Elnora, and siapa lagi? Maria and Michael, yeah. They use epoxy, yeah, in their report, right? Because epoxy can withstand even temperature as high as 95 degrees. The temperature that you need to use for denaturation of DNA. Okay, Ezra, any question further?
No, sir.
Is clear enough or disappointed
Yes.
that we don't have?
Enggak sih, sir. Tahu mahal itu.
It is
Makanya saya
expensive.
nanya.
Yeah. It is tens of million rupiah.
Oh,
I think it's
okay.
about 2,000 US dollar or something like that. And it's not economical. You know, we have the technology ourselves from batik technology, the technology built by our ancestors. You can use the technology. You don't need to make a digital wax printer. Alternatively, so that method, yeah, using paper is one way. The other way is using batik cetak like technology. Batik cetak, you make the cetakannya, the template using PMMA, or which you can use 3D printing, yeah, either acrylic or the plastic made from 3D printer. Then you use that to pattern the hydrophobic barrier in the fabrics. It's another way. But for wax, I have one of my students done using the old student. Unfortunately, it's not published. Using wax and acrylic. Okay. But we haven't never tried. We have never tried yet to use epoxy. You cannot use wax. Why? Because it melts. For PCR analysis, doesn't fit because it melts. Even when the temperature is 60 degrees, 40 degrees, it starts to melt already. So it's better you use a more heat-resistant hydrophobic barrier. Have you tried already for project sensor? Building something?
Yeah, yes, sir. But I'm still trying to make it nicer, rapi gitu.